Hello YouTube community. I hope everyone's doing well. Today I'll be talking about proper lab safety and good lab etiquette. This is a topic I specifically wanted to touch on because ever since I was a kid I was always very interested in science. In fact, I actually got my Bachelor of Science degree in biological sciences, so I know quite a bit about lab safety. I took AP chemistry in high school, back way back when, and I've done extensive lab courses in college, so I know all about lab safety. So let's begin. In life and in your laboratory, safety is the most important things. Mainly where the location of safety equipment is and how to properly use that safety equipment. Always be sure to note the locations of your overhead shower, the eye wash, fire blanket, first aid kit, and fire extinguishers. Obviously, in the lab, you'll likely be working with strong solvents and acids and bases. So use these chemicals in the hood only. Make sure your hood is also turned on. Your general laboratory should have a designated area for broken glass, and that's the only place it should be discarded in. There should also be a biohazard safety bin for certain proteins or bacterias, and you can probably find them labeled in your lab somewhere. This is the only place where biohazardous material should be disposed of. And last but not least, there should be a sharps disposal box within your laboratory. Any sharp items or pasture pipettes should be discarded in there. Now it's time to talk about lab etiquette. So when performing an experiment, there are two factors we want to consider: accuracy and precision. Now, for some labs, depending on where you are, precision is quite important, whereas for other labs, precision is not as important as accuracy. And the distinction between the two is that precision is how close each measurement is to the other, whereas accuracy is how close each measurement is to the actual value of whatever you're measuring is. On the topic of accuracy, there are a few things that most scientists know. For one, in general, accuracy in measuring volume is not very good for anything less than 5% of a pipette's total volume. And most of the time, the calibrations on a beaker or Lermeyer flask are only useful for approximations. They're not necessarily accurate. So now that we know how to ensure that our data is good, we need a means of recording said data. Most scientists prefer a notebook, usually a computation book with small grids. Composition notebook will do. And the general format follows something like this. We would include all methods on calculations or formulas used for calculation, the experimental conditions like the concentration of solutions, exact temperature, tabulated data, and comments concerning various steps of the experiment, and definitely descriptions of possible sources of error. The time in universities, you'll be expected to calculate all of the stock solutions for yourselves. Once you've collected the data, you can begin your report. The first part of the lab report is the purpose. It's a brief statement explaining the scientific goal of the experiment. Next, you need to provide the introduction, which is background knowledge for the principle explored in the experiment. A brief statement of the design of the experiment and the theory, including the summary of what you did, is all necessary for the introduction. Then you want to have another section which lists the materials you used in the experiment, including all apparatuses, chemicals, and safety considerations, including MSDS information. Next, we go to the method, where you summarize in your own words the written procedures you followed in the experiment. Now comes the data. Make a section for your results, and you can use several methods of statistical analysis to draw a conclusion. Be sure to include any graphs or normality checks in your results section, and label all figures. Lastly, draw a conclusion from everything given.